I've, the little bio set up there, I got a degree in chemistry at the UW, so I didn't go too far. And I worked about 35 or plus years at Sunstrand, starting out in the chemistry lab there, analyzing plating bath solutions because we built our own circuit boards and did our own metal finishing, and progressed into general materials and process engineering, which I used for many, many years of my life at work. So, but my real, I think about chemistry a lot because I think that chemistry, just like the last small phrase on the slide shows, I like the phrase that I learned in college, better living through chemistry. Because everything around us is chemistry, isn't it? Every part of what we're looking at here, every part of what we look at in the world is chemistry. Of course, physics is involved too. I love physics, but I love chemistry better. Everything's made of atoms and molecules and the interaction between them. So that's what's really good about chemistry, better living through chemistry. So the talk tonight topic is real chemistry doesn't support the origin of life by chemical evolution. That's what we're going to talk about tonight. Now, we're not going to get too deep into chemistry because it is kind of complex if you're not involved with it regularly, but enough just that we'll have a good awareness of it, and we're going to focus on issues that relate to the evolutionary concept of the origin of life from a chemical point of view, and how there's some real struggles with the model following real, actual chemical science. And so we're going to touch on a number of different topics in chemistry related to the cell and its function and the molecules that are involved in it. So I hope it won't be too complicated, but it is going to be kind of top level. So there are many, many issues that are kind of like that book says, the Achilles heel of evolution. You know, there's cosmology, and there's star planet formation, fossil record, Chris is going to be talking about that, genetics, Rob Carter is going to be talking about that, geology, natural selection, dating methods, the human spirit or morality and ethics and stuff. Where does that come from? Information science, but what we're going to focus on tonight is the origin of life, and that's going to be our subject and the most significant thing we're going to talk about. One thing that I have thought about science is that science is a very, very good methodology of understanding how things work, but it's very, very poor at finding out how things started. And that's very important because we're talking about the origin of the first living cells. I like this little comic or poster that I found, and it relates to science and how we view it, because if we're not part of the solution, we're definitely part of the precipitate. Does that make sense? So what is chemistry? That's the first thing we have to look at. Chemistry is a study of substances in terms of, one, its composition, you know, what things are made of, the structure, how those things are put together, and the properties, what characteristics does it have, and most importantly, the reactions between chemicals. How does it behave with other substances? One of the men that I really appreciate is a man named A.E. Wilder Smith. He's a Swiss fellow who has multiple degrees, and he made an interesting statement talking about the relationship of life and chemistry. And I'll read that here. The pure chemistry of a cell is not enough to explain the workings of the cell, although the workings are chemical. It's kind of like they're yes and no. It's involved in the chemistry, but it's not the chemistry. Does that make sense? The chemical workings of the cell are controlled by information that does not reside in the atoms or the molecules themselves. That's a really important concept to remember as we go through this talk. And when you think about how chemistry relates to the origin of life, there is information and communication, but it's not inherent in the chemicals themselves. It's just not, because they are just dead, inanimate chemicals. So, the model of evolution talks about the Earth forming 4.6 billion years ago. Chemicals form molecules from a primordial soup or perhaps an ocean thermal vents. There's a lot of different theories about how that happens. Molecules then bonded themselves together to make living cells. Well, it's basically formed by chance over time and the necessity, natural laws of chemical interaction. And that's kind of the concept used in the evolutionary concept. Like the little illustration on the bottom, the pictures, time plus atoms plus the roll of the dice equals life. Very interesting. I like this comic. This is one of my favorite comics that I ever ran across. This is Garfield's view of the origin of life. The very first cat crawled out of the sea about 10 million years ago. Unfortunately for him, it was only about another 5 minutes or 15 minutes before the first mouse crawled out. That's very interesting. I think that was very revealing. The author of Garfield had a lot to say about evolution in some of his comics, if you read some of his others. Here's a man named Frank Harold. He's a professor of biochem and molecular biology in Colorado State. He said, from an evolutionary point of view, life arose here on Earth from inanimate matter. Notice that word, inanimate. By some kind of evolutionary process. That's a kind of a special word, some kind. About 4 billion years ago. This is not a statement of demonstrable fact, but an assumption almost universally shared by specialists as well as scientists in general. That's an interesting comment, isn't it? That gives you a lot of confidence, doesn't it, in the chemical origins of life from an evolutionary point of view. It is not supported by any direct evidence, nor is it likely to be, but it is consistent with what evidence we do have. Well, okay. The model of creation that we look at for origins of life is a little bit different. For by him were all things created that are in heaven and on earth, visible and invisible, whether thrones or dominions or principalities or powers, all things were created by him and for him. So, the critical nature of origin of life and the evolutionary model. Well, it's biology's biggest challenge, actually. And many avoid this subject. One of the things that got me interested was a couple of years ago I was on a trip with a evolutionary atheist person that lives in this area. We were actually going over to the Dry Falls area, and we stopped and we had a discussion about evolution. And I asked him what he thought about the origin of life and how that relates to the evolutionary model that he was thinking about. And his comment to me was, it really doesn't matter how the origin of life came about. What's important in the biological scheme of things from an evolutionary point of view is natural selection and how once life is gone, is active, and then we have the progression through various species and on and on and on. But basically, he refused to discuss with me anything about the origins of life from a chemical point of view. I thought that was a very interesting comment that he made. Some argue that the origin of life is not part of the theory of evolution, which is not really logical, is it? It's kind of like a foundational thing. Considering the evolutionary origins are at best inconsistent and actually contradict real chemical science, and I think that's the reason why it's avoided. The origin of life really is a necessary precursor. That's a word in chemistry, is precursor. Something that you start with. It's the foundation of what you're going to do in any particular thing. You have to have a solid foundation. Whoops, I think I pressed the wrong button. The purpose of this presentation is to discuss evolution's most critical obstacle. Evolutionists are no closer solving the riddle of how life began And biochemists have been unable to show even the simplest cell could have been formed without intelligent cause. So there's a picture here of Mueller's experiment, which we probably have all heard about in the past, with attempts to form biological chemicals, which really didn't work too well. This presentation will examine the origin of life and illustrate that evolution is simply without scientific foundation. So, but first we want to talk a little bit about some terminology, because we're going to be using these. And you guys probably know a lot of this, but let's go through it quickly. Of course, atoms are the basic unit of matter. We get carbon, hydrogen, sulfur, nitrogen, nickel, iron, and other things. Molecules are specific arrangements of those atoms, like water, H2O. Amino acids are molecules used as the building blocks for proteins. And that's a simplified schematic of what an amino acid looks like. It has a carbon with a nitrogen on one end and what's called a carboxylic acid on the other, i.e. amino acid. Does that make sense? And then that little R group that's pointing straight down is anywhere from a hydrogen to other complex organic molecules that are kind of stuck on there. And those R groups are the groups that determine what the name or the actual amino acid is. There's 20 of them that are used in our biology. There are hundreds and hundreds of them in reality, out in the real world, but there's only 20 with 20 little different R's. Does that make sense? Okay. Proteins are just chains of amino acids hooked together in a string, linearly. Okay? So you have one kind, another kind, another kind, hooked together, and we're going to talk about that in a minute, how they are. This is a little bit more of a complex diagram. It's a diagram of a DNA molecule, deoxyribonucleic acid, which of course contains the basic genetic information. If you look at this one, it has a backbone, which is on the outside edges in the blue, the little pentagons. The deoxyribo with a phosphate group, you can see them kind of hanging off of one side of the little blue pentagons. Then on the other side, there's what are called the bases. Those are the colored ones, the pink and the green and some blue. There are 4 of them identified, 2 at the top and 2 at the bottom. There are 4 different kinds, and that is the alphabet of the code for the genetic information. I think most of everybody's familiar with that. Down at the bottom of this diagram, you'll see a DNA double helix with an expanded open area, and that is what's in this particular picture represents what's called a gene, which is a series or a section of the DNA that has all these different letters, those 4 bases in a coded sequence. And those are called codons. Each 3 letters represent a codon for a specific amino acid. So you can see if you have a long chunk of a gene, it has codons for many different amino acids in a particular sequence. Does that make sense? So that's how, when the DNA is read, then the proteins that are in our cells are manufactured based on that sequence as the reading goes down that gene. Okay? These are just, again, terminologies. And then the DNAs in our actual cells are bundled together with protein molecules to form the chromosomes. And here's a couple of examples of different amino acids and their appropriate DNA codes, like a U, a G, and a C. Codon means a cysteine amino acid. And this one, a leucine, serine, and aniline, for example. And there are 20 of those in living systems. Another molecule that we use, or that's important, is what's called a messenger RNA, and it has what's called a ribose, or it's a sugar backbone. That's kind of the ochre-colored or yellow-colored pentagon, and with a phosphate backbone attached. And then each one of those A's and G's and U's and C's are the codons that are copied from the DNA in the nucleus and then moves out into the cell to make the protein. That's why it's called the messenger RNA. It carries a message from the DNA out into the cell. You don't want the DNA out in the cell because things could happen. And then that particular messenger RNA meets up what's called a transfer RNA, which is another ribonucleic acid that transports the amino acid. Each transfer RNA is specific for a specific amino acid. So there's 20 different ones, basically looking at it. And that moves out, that's in the cytoplasm also, outside the nucleus. It meets up with the messenger RNA and acts as kind of like a truck carrying the amino acid to the site in the cell where the protein is made. So we got the messenger and the transfer. And then those two meet up in a little body called a ribosome, which is a large, that's the pink-looking glob there. You can see the transfer RNA in the upper part, and the messenger is stuck on the bottom part of the ribosome, and the transfer RNAs bring the amino acid in a particular sequence that's defined on the messenger. But it's at that particular site in the ribosome. I just think it's kind of interesting that the ribosome is made of proteins too. That's a question. Which came first, the ribosome or the messenger RNA? I don't know. Because you got to have the ribosome to make the messenger RNA in the first place. So what we're going to do from now on, those are some of the terminologies. I know it's fast, but hopefully you know a little bit about this anyway. So we're going to talk about some of the problems. And these are the kind of things that they're in no particular order, by the way, of importance. They're just a set of problems chemically that we have to think about in relationship to how life formed originally versus how life is operating in the cell. One of the key things to remember is that chemically in the cell, these processes are protected chemically from random chemical reactions that could interfere. Out in the open, in the pond or in the ocean, there's no protection for these chemical reactions we're going to be talking about. And so there's problems in an open environment that you don't have inside the cell. Does that make sense? It's a protected environment there. It's set up to do the job it does, and it does it very well. Out in the open ocean or pond, it's open to a lot of other chemical problems. One of the things is basically the information problem. Information is everywhere, isn't it? Does chemical information or evolution, time and chance, align itself with our knowledge of information science? You've heard people say over a long period of time, by chance, all these molecules happen to get together, correct? In the right order, that would be biologically active. Well, what is chance? It's very similar to natural selection in the problems. One, chance is not a thing or an entity, is it? Have you ever thought about that? Anybody ever say, oh, this happened by chance? Anybody ever say that? All of us have done that and said that. But really, is chance a thing? No. It's an active force? No, it's not an active force. It's not an agent of causation. And that's something important to remember. It is simply a probability of something happening. It's an accidental event. It is something that happens by accident or without design. It just happens. It's more, I think the best definition is just a probability of something happening, like throwing a dice. Have you ever thought about what causes a particular number to come up on a dice throw? Is it chance that causes that? No, it's not. It's how you throw it, it's the weight, it's the twist in your arm, or whatever, many, many causes physically that cause something to happen. So what is not information? There's a lot of things out there. Is order or data or organization data? Is that information, I mean? There's a picture of a cube of ice, or excuse me, a cube of salt. Is that information? Or is it just order? It is just order. Sodium chloride molecules in a matrix. Even the series of amino acids hooked together in a protein is really not information by itself. It's just a set of data. It's an organized set of data. How about a snowflake? Is that information? No. It's a really specific order of water molecules. What is information? Well, information is a code, it's a language, it's a knowledge or an alphabet that's sent, received, and understandable, or more importantly, executable. You have a sender and you have a receiver. It's the whole system. There's something that's transferred from one position to another, and it has meaning to it, that something or someone can take action on. So useful information, it's the ability to read and interpret and execute the code. That's what information really is. It's not a crystal of salt or a snowflake, although there's data there and there's organization. It's functional, it's useful, it's practical, and it's instructional. Here's a picture. Anybody recognize that? Okay, is there data there? Sure, you got red and yellow and green lights, don't you? But is that information? Red and yellow and green lights. What makes it information? Us. We know what the rules are. We know what red and green and yellow mean, don't you? I hope you do. Because it's very critical for what purpose? To keep us safe on the road and give traffic order and semblance of civility, right? We've never run a red light, have we? Okay, what happens? Wreck, okay? We know what the result's going to be. So the stoplight itself is not information. The lights are not even information. But how we interpret and use the language of that light makes that system. That's what the information is. There's a sender and there's a receiver. That means there was the stuff, the light, there was a code or a system that was set up, i.e. the laws, and then we understand what they are and we can apply them. That is information. But the individual things are really not. It's just data, just order. The cell is full of prescriptive information. The DNA and RNA and proteins and other cell molecules execute cellular action, resulting in meaningful and functional output. And there's a list of the different items on the left-hand side that it accomplishes. With that string of protein molecules or with the string of the DNA bases in a particular order, which is just data, it's not really information per se, but there's a prescription inside the cell that understands how to use that, how to execute that. So that makes it a system. It's a system rather than just pieces of data. Remember we talked about the fact that atoms and molecules by themselves are not an integrated information system. It's not like our computer. It wasn't there. So think of those molecules out in an open ocean, floating around by themselves, that are inanimate themselves and have no program with them. They just have pieces of bits of data. They're not useful information. That creates a problem for the evolutionary concept because we look at the cell and it's full of prescriptive information. So how did it translate? How did it get to that point? And the actually only way it really works is because the cell is a kind of a unified biological computer with a code and a system for translating the data that's there into useful information. Does that make sense? But out in an open pond, it doesn't work that way because it's just data with no prescription to it. Where did it come from? Well, I think this is one of the most important things that we want to talk about tonight, as well as the other things. But Dr. Wilder Smith calls this thing that rides on chemicals, he uses the word teleonomy, which is a kind of a strange word. We don't hear about that word very much. It comes from two Greek words meaning end purpose and law. It means end directed and purpose law. It's the active operation program or force in living systems. It's more than the information or the data or the little bits of data or any order. Does that make sense? It's not physical, but it's metaphysical. It's problematic and computational aspects of purpose. And it's the quality of purposeness. Not purposelessness, but purposefulness. Goal-directedness of structures. For example, on the top, on the top here, we have a picture of some trees, right? If you look at the bottom, we have the same trees. What's the problem with the bottom? They're dead. Okay, what's the difference between the top and the bottom? It's the same chemicals, the same available energy that's around. Actually, the same information is there because it has DNA in those dead cells. What's missing? Teleonomy. There's something else besides the stuff. Something else even besides the information. It's beyond the information that causes life systems to work. Here's another example. I like this one. I grew up in a farming area in eastern Washington. The chemicals and the information are not enough. If you wanted to raise corn as a business with high quality yield, what would you need? Well, what's the cause of accomplishing that? First of all, you're going to have stuff. You're going to have farm, land, seed, equipment, fertilizer, water, sun, etc., correct? That's needful, isn't it? That's like little pieces of data. In and of themselves, they're by themselves and really don't mean much. Then you have to get a little bit higher complexity here. You have information now. You have knowledge of the farming techniques and timing. But is that going to result in, you can read a book, right? You can go to Wazoo and learn about it, right? What's missing? Anybody have an idea? Teleonomy. You know what teleonomy in a farm is? A skilled farmer that can actually execute the plan with the stuff. Does that make sense? Without the farmer, all this stuff is really insignificant in terms of having the result we want. Same thing when it comes to chemicals floating around in an ocean. You can have the information even. You can even have the proteins there. Maybe you could even have the DNA there. But there's one other thing that's missing, something to organize it. The farmer is outside the stuff needed and knowledge required, isn't he? But he puts it together. He uses this stuff to activate and implement the plan, okay? So the cause of life, the stuff, atoms, molecules, laws, energy. The information, we have the DNA code, correct? The information is there, the data in an appropriate manner, but we need a skilled source that actually executes the plan. What or who is that skilled source? Anybody have an idea? Remember, chemicals are inanimate themselves. Well, I remember this, don't you? Obi-Wan, when the Death Star killed Alderaan, he said what? He felt a disturbance in the Force. My wife doesn't like Star Wars because she thinks this is not exactly consistent with the Scripture or with God. It sort of paints a different picture. But I think it's a good illustration. Whoops. The real source of life. There's some verses. Then the Lord formed man out of dust and breathed into his nostrils the breath of life. As you do not know how the Spirit comes to the bones in the womb, so you do not know the works of God who makes everything. He is before all things, and in him all things hold together, upholding the universe by his word of power. See, that's what the creation model believes is the teleonomy that takes the stuff of nature, physical things, and makes it into living systems that work well. Because in and of themselves, there's nothing to drive them in that direction by themselves. Effect on the evolutionary model. This is a serious issue no matter how matter and energy arose out of nothing. That's the first bottom line, right? The prescriptive information did not originate or was generated by the chemicals and processes. That is not the source. A skilled source is required to initiate and maintain life processes that does not reside either in the stuff of life or the prescriptive blueprint of life either. And that's, I think, a really important issue to think about. How does this affect the model of origin of life from an evolutionary point of view? Well, I think perhaps maybe the model should be modified, or maybe we should abandon it because it doesn't provide that teleonomy, the organizing force. Well, that was a long talk about one little tiny subject, but it's a very critical one. It gets worse from a chemical point of view, and now we're going to get into a few other things. The law of thermodynamics, second law. Energy goes from the state of usable energy to a state of less usable energy for doing work. Complexity, it decays into disorder. That's kind of the name of the game. Regarding chemical origins, 4 necessary conditions to stop second law from hindering the formation of simple to extremely complex biological molecules and systems. Because think about that, we're taking the simple atoms and molecules that are floating around randomly and we're organizing them, bringing it up in complexity. And in an open system, that's a little bit difficult because it drives just exactly the other way. That's generally what the law says. So we have an open system. These are the 4 necessary requirements, at least from my point of view. An energy source. We have to have a way to capture and store the available raw energy, and an energy conversion system once you have the energy. The problem is outside the cell, and this is important, and before existing cellular processes were in place in the ocean or primordial soup, we didn't have number 3 and 4. We had the energy, we had an open system, but we really didn't have a way to capture and store things, the energy. and focus it to build the proper chemical processes and maintain them. A fundamental that science has never been able to solve is how to produce energy flow through a system to do this work of coding to produce, for example, a functioning protein. Living systems do, of course, harness energy for this purpose, but only because the required purposeful assembled metabolic pathway is already in place and functioning. In other words, life comes from life, doesn't it? Because it has to. Because that's the only place where the teleonomy is. It gets worse. Anybody heard of the word hydrolysis? You know what that is? Some of you have, maybe? Yeah. That's a chemical process. So where and how did it start? The life systems, the proteins. How about a primordial pond? How about pond scum? That's sort of a picture of what the evolutionary process thinks happened. On the left-hand side, life formed from the chemicals, and then, of course, you have the natural progression in the evolutionary concept. But from a chemical point of view, the ocean and the primordial pond is the absolute worst place in the world for a protein to start, or for that matter, a DNA molecule. Does anybody know why? Well, because proteins are formed when amino acids bond together during a dehydration process. If you take it to look at the top, you got two amino acids, and they're going to form what's called a peptide bond. And when they do that, one of the products is water. See in the middle arrow, the little water molecule that's given off as a product, and down below is the start of a protein molecule. That's where you are dehydrating the amino acids and removing a water molecule. Unfortunately, it's a reversible reaction. It can go naturally the other way. Now think about that. If you're in a pond of water or an ocean, what is going to happen to a protein peptide bond that just formed? It's going to break apart again from the word hydrolysis, the reverse reaction. Now, in an uncontrolled, unprotected environment, that is what will happen. And so you have proteins forming, and then they break apart. And they form, and they break apart. Does that make sense? This is a bad problem for an origin in a water environment. Now, in the cell, it's a watery environment also, isn't it? Right? But it's protected by enzymes and other proteins. And it thwarts the dehydration process as the proteins are made. It's a very controlled process in a controlled environment. It's kind of like a biological, test tube. Does that make sense? Rather than an open environment. So the hydrolysis problem is a real problem. And I would challenge, I think that's one of the things we can remember from this talk is that if you want to talk about a biological or a chemical process that's a real bugaboo for the evolutionary process in a water environment, hydrolysis is a big one. But it gets worse. There's more. In a environment that has oxygen, and this is a very complicated process, proteins and large chemical molecules are vulnerable to attack by what are called radicals, which are atoms that have electrons either stripped away or added to it and make them very, very active. In our environment, they are around and they do affect chemicals that are present. And it's not a good plan if you have oxygen around for those large molecules. But, on the other hand, we need it for respiration. The good thing is that the cells again, are in a protected environment and have that in a very organized process. But it gets worse. And again, these are just random chemical processes that are a real problem for a random origin of chemical evolution. So stay with me on this. These are a list of different things now. Once a protein forms, it has all kinds of these little R groups hanging off. Remember, that's part of the amino acid. And as it begins to be generated in whatever environment, they would naturally have a tendency, these R groups, to interact with each other, and the protein will start to fold up in different ways. Okay? Does that make sense? Hydrogen bonding, chemical reactions, other chemical relationships between the R groups in the little individual amino acids. And so it will fold up into this way. And in the chemistry of our cell, folding of the protein is absolutely critical to its biological activity. If it's not folded right, it will be non-functional even if it's there. So in an open environment, it's totally uncontrolled. It will fold randomly. Even if it forms, it would probably not be biologically active because it's folded wrong. Are you with me on that? Here's a picture of a simple amino acid over on the left-hand side, and you have 4 different levels of folding that go on to make a biologically active protein. And it gets more complex as it goes. I don't think we have to get into the details. It gets worse. There's other problems. The interaction with the general chemical environment. Unprotected amino acids, proteins, DNA, and RNA, and other biologically active molecules can react randomly with other chemicals in the primordial soup environment, destroying or deactivating the molecules or preventing their formation in the first place. They form and then they start reacting with other chemicals that have nothing to do with biologically active chemicals. And it will either destroy or modify the protein or the RNA or the DNA that's forming. That's if you do it in an open environment. In the cell, it's what? It's a closed system. It's very controlled by the processes of the cell itself. Self-protected, if you will, from the environment. But in an open environment, that's just like dumping a bunch of chemicals into a beaker and sloshing it around. You're going to get all kinds of weird things. That's the Mueller experiment, for example, where they did that inside even in a chemical laboratory. They got a bunch of organic junk, let's put it that way. It gets worse. In a physical open environment, we have heat and poisons and radiation from the outside, UV light. Anybody ever get a sunburn? What's happening to your skin cells? They're being damaged, aren't they? Okay, by an over-intense amount of UV light. Imagine a freshly made protein floating around in an ocean environment being attacked by one or more of these items here. It's sort of like holding a gun to it again. Not by other chemicals, but by just simply radiation, for example. It gets worse. Concentration problems. In chemistry, rates of reaction are very important. You have to have chemicals in close proximity to each other to actually react with each other, correct? Think about random chemicals floating around in an ocean. It's a pretty big volume, isn't it? Or a large pond, or whatever. In order to get a realistic number of chemicals to react with each other, they have to be a lot of them in close proximity. So concentration is a real problem in a natural environment because it's not concentrated enough to get enough chemicals to react correctly in the right way, even though it's randomly controlled. Inside a cell, very concentrated, very purposefully driven together by the cellular action. But how did it start? Because it was in a dilute condition before. It gets worse. Amino acids come in two different forms. If you have a specific, like leucine, for example, it comes in what's called a left and right-handed version. And you guys have heard about this before, haven't you? Take your hands and look at them. Left and right-handed. I did it right. How about that? Try putting them together. I don't think you can exactly get them to match any way you do this. It's just not going to happen. Okay? So it's a real problem. Now the interesting thing is that in the natural pond or the ocean or whatever, when amino acids are formed, they come both in left and right-handed versions, generally in a 50-50 mix. And when they react together equally from a chemical point of view. So when a protein forms in the natural environment, it's a mixture of left and right-handed amino acids that are hooked together. But if you look at the proteins that are in a cell, they're all what? They're all left-handed, 100% of them. Now how could that happen if you start with 50-50? And they react equally? This is a real problem. This is one of the it's a real simple problem, but it's a real problem. Okay? You understand that? So, in environment we have left and right-handed equal, and they react equally to each other. So they will form proteins very quickly, or a peptide bonds. But they're the wrong version. They're left and right-handed. And that's a real problem for the chemicals because when they fold, if you got mixtures of left and right-handed amino acids in a protein, then it will fold wrong. It won't be biologically active because the R groups will be at different sides. Sometimes they'll be like this and sometimes they'll be like this. It's like, and when they get strung out together, then they can't fold together in the right way. In the cell, in that controlled environment, only left-handed amino acids are incorporated into the proteins. Do you know why? Anybody know why? Guess what? The DNA molecule is all right-handed. You remember those ribose with the bases hooked onto them? They're all right-handed riboses or deoxyriboses. All of them. That's interesting. By the way, those occur naturally too, left and right-handed. But they're all right-handed, every one of them. And that kind of makes sense because when it's copied, it generates all left-handed amino acids. Make sense? And that's exactly why you have only left-handed amino acids in proteins. But the problem is, in an open environment, either one of those large molecules will have both. And the problem with DNA is if you're reading down the DNA and the DNA has to split apart, the double helix, and then it's read and then the messenger RNA is generated, it's a good thing they're all right-handed because if it hits one left-handed sugar in the DNA, guess what happens to the process? Stops like that. It's an error. And you cannot produce any proteins. You can't make enzymes. You can't make anything from that DNA. God was pretty smart. He made it all right-handed so that it could be read consistently. But it's in a controlled environment of the cell. But naturally, in the open pond, it forms randomly, left and right-handed, and it just doesn't work. So the question is how did the cell form with only, and think about that, how many units in a DNA molecule? Hundreds of thousands of units, and they're all right-handed. And just one of them would screw up the DNA. So how could that form naturally? The odds of that are really tiny. It just wouldn't happen chemically. So it's a real bugaboo for the kind of the random DNA process of making a cell. Okay. Yeah, we just talked about that there. Now, not outside the cell for sure, it's not going to happen. But inside the cell, yes. It's a definite yes that this happens because it's programmed to do that, and it's set up to do that. But it didn't happen naturally. It will never happen naturally. So how could it happen in the first place? It gets worse. There's other things. We have a few more minutes. This is going to get complicated. I'm going to go pretty fast. Another issue is what's called irreducibly complex problem. This is an example of the metabolism inside the cell. This picture is the first step in metabolism. When you have energy source, glucose, it's broken down because our cell needs energy to use, right? Okay, correct? Without energy, if we don't eat, and there's no energy source there, we will what? Die. The cells will die. So this is a very complicated process, and this is just a top-level view of it. It's even more complicated than this. So you take glucose right in the middle there, and it goes through a series of about 10 proteins to get to a precursor called pyruvate. Okay? Then it goes to a next cycle, which is called the Krebs cycle. And this is a circular cycle. If you look at the upper left-hand corner, you see a molecule, and that's the result of that step one. And it goes through another series of 10 or so chemical processes. And by the way, each one of those processes is performed by an enzyme, which is a what? Protein. Okay, so we have about 10 proteins involved in this cycle. Now again, we're just trying to generate energy. We're not there yet. Then the last step is called the electron transport chain, which is done in the mitochondria. By the way, a lot of this is done in the mitochondria in the cell. It's a 4-step process. That's those little blue globs there, which are proteins. And at those stages, you get 4 more chemical reactions, and you eventually end up with a compound called ATP. If you look at the lower right hand, you see a little circle that says ATP. That's the energy molecule for the cell. So it took, about 25 enzymes through this whole process just to convert our food into energy that the cell can use. And that's the way it operates. Do you think that's pretty complex? I think it's real. I don't understand. It is really complex. And this is just a little top-level view of it. There's an interesting little fact. Anybody hear of cyanide poisoning? When I was at the Sunstrand, I watched a woman die from cyanide poisoning. She committed suicide in our lab, right in front of us. It took about 5 minutes. She drank some coffee with potassium cyanide in it. 5 minutes later, she was dead. And do you know why she was dead? Because the cyanide, it interferes with that's where that X is on here, interferes with the next to the last step in the electron transport cycle. It deactivates the enzyme in the 24th step of the process. That's all it does. And guess what happens to the cell when that happens? It dies. Now, think about that. You got these massive complex enzymes in our system, in our cell, and every one of them is critical. If you interfere with one of them, the cell, what? Dies. Think about this happening by random in an ocean or pond, how all these things got organized. How many of those processes would have to be present in order for our current system to be working properly? How many? All of them. At once. That is hard to imagine. It happening outside a controlled system, isn't it? Fortunately, inside the cell, it works really well because it's controlled. Does that make sense? That's a real problem, in my opinion, for random chemical evolution. It just doesn't make sense. Cellular complexity, irreducible complexity. All the protein, enzymes, and other required reactants, a functioning complex mitochondria, cellular organelle, and other cellular structures, and even the end product ATP energy unit are all required to be placed in place. By the way, you remember I said the end of this process is the ATP, right? This long process. Guess what? The ATP had to be there in the first place for all this stuff to work, because guess where the energy comes from that the enzymes use? In those 24 enzymes that make ATP, guess what? They have to have ATP to work. Isn't that sort of like a chicken and egg problem? Which came first, the ATP or the enzymes? Well, you need the ATP to make the enzymes, but you need the enzymes to make the ATP. This is a problem, isn't it? Well, here's an example of another one, protein formation. You have the double helix there. You have a enzyme called RNA polymerase, and it breaks apart the DNA and makes the messenger RNA, and then the messenger RNA goes out and makes a RNA polymerase. Well, which came first, the polymerase or that's a problem, because you need the polymerase to make the polymerase in the first place. Does that make sense? It's a chicken and egg problem. I'll skip this. This is another complex issue because our time is ending. A probability problem. Anybody ever flip a penny? Here's some data that show just generally what the probabilities for getting a heads every time is, depending on how often you want to get a sequence of heads. That's pretty hairy, isn't it, when you want 100 in a row, right? Well, think about amino acids that are 150 amino acids long, and they only can function if they're in the right sequence. 10 to the 164th power chance of that happening by accident. Do you know why it happens in the cell so well every time? Because it's controlled. It's pre-established. There's a code that defines that. In pond scum, you got a problem with probability. Well, where does this lead? I mean, those are just some examples of chemical processes that cause a problem for the evolutionary concept. Dr. Wilder Smith made this quote about Einstein. Even though he was not a Christian, Einstein concluded that the universe and therefore biology must have its origin in understanding, thought, and concept, mathematics, intelligence, and teleonomy. Oh, that's an interesting word. And not in randomness, chance, plus the inanimate laws of nature. It just doesn't happen. For to him, Einstein, the greatest miracle in the universe was that we can at least in part comprehend it. That's pretty neat. We're pretty smart. We know a lot, but we don't know very much. But we know for sure that the chemical laws really run counter to the evolutionary chance of things happening. There's another viewpoint. Dawkins, you've heard of Dawkins? I'll go back to that. He said that you're either ignorant, stupid, or insane if you think that evolution doesn't work chemically, for example, by nature, by chance. Well, what does he really think about the origins? He was interviewed by Ben Stein, and I actually watched this in the video called Expelled. Stein, how did life start? He asked the same question I asked that evolutionist when I was on my trip. Richard Dawkins, who told us that we were stupid if we believe in, a miraculous God who did it. I have no idea how it started. Perhaps it was seeded on Earth by a higher alien intelligence. That's a quote. That's what he said to Stein as an answer to where life came from. Well, the aliens did it? You got to be kidding. Well, what does it mean? Well, I think it's important that we understand that it can't work by random chance. And it's really dependent on life first originating by natural processes that really violate basic chemical laws. The entire model of evolution is without a foundation because it just violates them. This is plain and simple. And I like Mike Riddle's quote here. Why should I accept evolution when you cannot produce the evidence? Because there is no evidence for a chemical evolution. I already have faith. Tell me about your faith, and I'll tell you about my faith. Therefore, I think it's important we understand who the life giver is. And that's the basis of our Christian faith, isn't it? It's reasonable to believe God did it, because that really answers the question, how did it happen? Well, our time has kind of fleeted by, so I'll just remind you that this is why we need to at least be a little bit prepared, even in some basic things like left in hand, right. If I were to take two things out of this whole talk, I'd think, one, left and right-handedness of amino acids, that isn't going to work. Hydrolysis is a problem. And the question is you can have all this stuff there, but what drives it to work together? What is the cause? It's not in the chemicals themselves. So they can't do it naturally in an open environment. It has to be from the outside. Well, in the evolutionary world, there is no outside. Does that make sense? There is no outside source. I think we can finish now. I like this one. This is my favorite, some of my favorite verses. Ever since the creation of the world, his invisible nature, namely his eternal power and deity, have been clearly perceived in the things that have been made. So they are without excuse, and exchange the glory of the immortal God for images. Whoops. So we're done. If you have some questions, we have a minute or two, right?
We do. We can take a few questions. Let me bring the mic around. And we still got about 10 minutes, so make them good.
I just realized you didn't cover about timing, how timing is necessary for processes to actually work. Like, for instance, chemical processes require that two things be there at the same time, for instance.
Yes, and that part of that has to do, remember when I talked about the concentration problem? In order for chemicals to react, I remember in the chemistry lab, we had to be very careful how the sequence of adding chemicals to end up with a proper reaction or the results, but we also had to carefully control the concentrations and how we added them together, what kind of energy we supplied to the system, like a hot plate, or sometimes we had to put catalysts in the reaction chamber to have something done. So concentration and sequence of events is very critical to get the right result in a chemistry reaction. In an open system like an ocean or a primordial soup, there's nothing to control that. And that's why you get all kinds of cross-contaminated reactions happening. Concentration effects, sequence effects, and the types of chemicals you're using. You have to be very careful what chemicals you introduce into the system. They can kill the process. Does that make sense? So you're right. Timing is very important. I don't know if that helps.
Any other questions? Still got a couple minutes. Thank you, Ron. Appreciate that a bunch.
Okay.
Give me just a second. I'll let you close this out in a word of prayer.
Yes.
Remind everyone that next week I'm going to be doing a talk on dinosaurs, which, again, if you don't know how dinosaurs fit into the biblical history, could be very eye-opening. And Robert Carter's coming up. Was that 28th at Arlington, up in Arlington at the Atonement Free Lutheran Church. Again, Robert Carter is down from Creation Ministries International, and he's apparently going to be here for about a week. So I believe if you went to, so the Apologetics Forum of Snohomish County, if you go to their website and look at the event announcement for their talk with Robert Carter, I'm sure Heinz has all of his talks listed there. And so, again, this is a geneticist who's here from Creation Ministries International, one of the big creation organizations internationally. So, good opportunity to hear some talks by someone who really knows the science. Again, a PhD in genetics, so he can really talk to the information science aspect of this. We have an offering plate in the back if you want to help us. Those the monies, those checks go to should be made out to Cedar Park Church. All this money gets turned over to them, but is used and dedicated to pay the honorariums for this specific program. So you could help us on the way out. There's still some coffee in the back, and there's a bunch of books and videos, including a few back there on the origin of life, if you want me to direct you to those. And then there's a sign-up sheet for Northwest Creation News if you want to learn about upcoming creation events. Ron, I noticed you had a slide there about upcoming field trips. Do you have anything on the book yet?
Not yet. Let me go back to that.
Ron does field trips every summer out to both Mount St. Helens and the Channeled Scablands, which
Yes.
Channeled Scablands is a very, very important historical event where Washington was destroyed by a major flood, but the geologists refused to accept it for about 50 years. Because they're just highly biased against catastrophic flooding as an explanation for Earth's geology. And so it took them about 50 years before they eventually came around and said, okay, I guess those features in eastern Washington were the result of a flood.
And that's a lot different in Mount St. Helens because we actually photographed and were present for each of those.
That one, yeah.
But it does have still a message, very similar to that.
So no dates yet.
Not yet.
But
I usually do those, like in June and July and August. Mm-hmm. But I do open up for anybody that wants to go anytime, because I'm retired, so I am available. I will drop anything and go to those on a tour. So just get a hold of me anytime.
Well, let us know when you get the dates, and
I'll
we'll make
have
sure
those
those
dates
announced.
shortly. Yes,
Yeah.
yes.
Okay. Sounds awesome.
One thing I wanted to share as we close in a word of prayer too,
Yes.
and thank our Lord. 2 verses really are important to me. Psalm 118:8. It is better to take refuge in the Lord than put confidence in man. Because even when people know that the chemistry runs totally counter to evolutionary origins, they still lean on those things because there is no option for them. Otherwise, you have to let in the cause. Okay? And they don't want to understand what the cause was. I can understand why that evolutionist atheist did not want to ask or talk to me about origins because he had no answer. He had aliens on his mind too, I guess. The other one that's really cool is Psalm 111:2. Great are the works of the Lord. Now this talks about the general works of, other than just, physical creation. It talks about all of his works, but this is included, what we just talked about. God is the greatest chemist in the world. There's never been anybody better. And great are his chemical works, studied by all those who have pleasure in them. Chemistry is a pleasure to learn about, because it speaks about who God is. And that's what I'm really excited about. So let's thank him. Thank you, Father, that you are the great chemist. Thank you that you had us in mind when you created chemistry to run this world by. And all the other physics and everything else. You just put it all together in a wonderful fashion. And thank you for the cell that works so well and keeps us alive. Oh, Lord, thank you that you breathe your spirit into us so that you can take the atoms and molecules and proteins and all those things and let them flourish in our life so that we can live and understand who you are. You are the great King, and we just thank you for that. In your name, amen.